My name is Erin Talbert. I'm an assistant professor at the University of Iowa, and it's my pleasure to chair your session this afternoon and to introduce our first speaker, Bert Blaauw from the Veneto Institute of Molecular Medicine, who's going to tell us about muscle tumor crosstalk during cancer cachexia.
All right, so I will give you a bit of a talk on the project we have ongoing in the lab, which will focus on the regulation of mass and of the muscle and how this actually can affect also the tumor. So, of course, this is a well-known image where you know the cancer cachexia is obviously a systemic disease affecting many different, if not all, systems in the body, and we are focusing in the lab on the skeletal muscle. And so this is a standard slide I use for many different presentations, also when I'm not talking about cachexia. But here, actually, you can see I think I will try and convince you that the systemic role of muscle, whether it's through as an endocrine organ or through the regulation of body metabolism, could actually have an effect also on tumor growth. Okay, so muscle is a very plastic tissue. So here, this is a very simplified scheme. So it can, from its, let's say, basic condition, either get smaller or bigger, and this depends really on this balance, whether it's pushed towards protein synthesis or more towards protein degradation. And this is regulated by these two different systems with regards to protein degradation, so the ubiquitin proteasome system and the autophagy lysosome system, which I think more or less make up about 90% of protein degradation. And on the other hand, you have protein synthesis. And so here there are multiple pathways regulating this increases in protein synthesis. And the one we will be discussing a bit today is this downstream of IGF-1, PI3 kinase, AKT pathway. So here you see that there are, for example, here we have two major regulators, I think, of adult muscle mass, these two hormones, IGF-1 and myostatin or follistatin. And both of them, through different receptors and different signaling pathways, actually go through AKT and mTOR signaling to actually modulate protein synthesis and is quite important in stimulating, for example, muscle growth. And a lot of this is, let's say, the central role of mTOR signaling. This was already done many years ago also by David Glass, who's here in the audience, and was done by using rapamycin, this specific inhibitor, to actually block mTOR to underline its importance in this growth stimulus. And so since it's so important in modulating and regulating adult muscle size, so we wondered during cancer cachexia, how does mTOR signaling change in the skeletal muscle? And so we use this C26 model, which you can see here. So we've seen this already a couple of times pass by. So we monitor this with the echo MRI, and so you can see very nicely, and in our case, more or less, I saw some other numbers before. We see around day 10, these mice start to lose body weight, and this is due to a loss in lean mass, but also in fat mass. And then when they lose around 15 to 20%, we sacrifice the animals. This is kind of our endpoint. So the muscles get smaller, and actually if you measure the muscle force, you can see also that they actually become a little bit weaker as well. So it's not just a loss in size, it's actually also some, let's say, defect, some intrinsic defect in the mechanisms leading to force production. And when you look here at this tetanic force, which we generally measure in this in vivo model, you can actually see that we have a significant reduction in tetanic force production. Good, so in these muscles at the end, let's say when we take them out, so when they lost about 50% of their weight and they are actually functionally compromised, what is happening to the mTOR pathway? And so this is an extremely simplified scheme. So this is a very complex, let's say, signaling pathway, and we focused on these two downstream mediators, so S6 kinase and 4-EBP1, to see if they are changed. And here there are some plots, and I'll, as you can see here, for example, we see this reduction in ribosomal protein S6 and also some reduction in the phosphorylation of ribosomal protein S6, and also an increase in 4-EBP1. And this is an inhibitory protein, so this actually binds to this 4E protein and therefore inhibiting cap-dependent protein translation. So if this increases, this actually reduces protein synthesis rates. And so here we actually quantify this. So we really see at least some good indications that in this cachectic muscle, mTOR signaling is down. And then we also wondered, so this is nice, it's a good indication, but does this also match to what happens with regards to protein synthesis? And so for this, we use this puromycin assay. So you just inject puromycin, and you see this incorporation. And there we could quite nicely see, as you can see here in the cachectic animals compared to the sham-treated animals, how the incorporation of puromycin is significantly reduced to these cachectic animals. So we have a reduction in mTOR signaling, and also, let's say, it's accompanied by a decrease in protein synthesis. Okay. All right, so now then the question becomes, so if we actually restore, mTOR signaling and therefore most likely also stimulate protein synthesis, can we then actually revert this, this muscle wasting? And so to do this, we used, actually go a little bit further upstream, so we go to AKT, and we use the mouse model where we can actually overexpress this myristylated form of AKT only in skeletal muscle. And it's nicely time controlled. So when we inject tamoxifen, it binds to the estrogen receptor domain. AKT goes into the membrane and starts to phosphorylate its downstream targets. And already now, many years ago, we published this paper where we saw that, I don't know if you can appreciate, but the muscles become actually much bigger quite quickly. And if we wait about a couple of weeks, they don't only get bigger, they actually also get stronger. So it's really quite a nice model, at least in a relatively short time frame, to see a functional increase in muscle mass. And so the scheme, the experimental scheme is the following. So we take these adult mice, we inject the C26 cells, we wait till they lose about 10% of body weight, and then we start to give tamoxifen. So at this time point, we activate AKT, and we want to see if we can rescue, in this case, body weight. And as you can see here quite nicely, following the green line. So when you see here the gray one, our tumor-bearing animals, they actually lose body weight continuously, as we would expect. But when we activate here AKT, we actually see a nice recovery, at least partial recovery here, of body weight after activation of AKT, and this happens in about a 5-day time frame. So when we then look at lean mass instead of just body weight, we can actually see that we have a complete rescue of muscle mass. And here, when we actually take out the muscles at the end, you can see we have a very nice rescue in our cachectic animals. They get, they have their atrophy, and this is completely rescued and even grow a little bit bigger after activation of AKT. And this is not just an increase in size, which in a way, I guess, was for me not extremely surprising. So there's not a huge anabolic resistance, if you want. So this was perhaps somewhat a nice result, but also histologically, they look very nice. And then we looked to see some other functional properties in these, let's say, rescued, reverted muscles, and we looked at some metabolic aspects. So here, for example, we checked the glucose uptake, and we did this initially here, just looking at, this is a pus staining, so we look at glycogen content. And so in cachectic animals, at the end, we actually see quite a significant reduction in glycogen content. And when we activate AKT in these muscles, we actually really pull in the glucose and we restore glycogen levels. And it's actually even higher than it was before the cachexia. And this is, of course, due to what we did this deoxyglucose assay. We just inject this into the mouse. And you can really see that in the tumor-bearing AKT mice, we have a very strong increase in glucose uptake, explaining what we found with the glycogen staining. So is it only histology? Well, it's not. So now we went back to this functional evaluation, the in vivo approach, which actually the Aurora people are here. So you can check out the setup we use. And what you can see quite nicely here is that, so this is an absolute force. So you see that the cachectic mice, which actually become smaller, they have the smaller muscles, they of course become also weaker. And if we then activate AKT, we get this regrowth. So keep in mind that the muscle did become smaller and then it comes back bigger again. Actually, we get a complete restoration of muscle force. So this is actually, for me, it was quite nice, quite surprising, because this is in a 5-day time frame is quite, I thought it was quite impressive to get this functional regrowth of the muscle. And so how to explain all this? So how does AKT do this? And so to do this, to understand a little bit better what is going on in these muscles, we did this, this is just a bulk RNA sequencing, but this was for us already quite enough, I think, to get a good idea on how strong the effect of AKT is in let's say, normalizing a lot of the muscle properties in these mice. Because when we look here, we see a principal component analysis, and if you look at the small circles, you see that the cachectic animals, so these are two different groups, but they all kind of cluster together. And here, the very small ones, these are the sham animals, they cluster together at another site. But when we activate AKT, these big green circles, in a tumor-bearing animal, we actually restore, let's say, our transcriptome in the muscle to very similar to what you see actually in a wild-type animal. So this was quite surprising. And here you see this in a heat map. I think for me this works a little bit better to understand. So you see the cachectic mice here. You see how there's a whole cluster of genes which actually increases very significantly in cachectic muscles. But when we activate AKT, we actually see a complete, let's say, normalization of a lot of these genes which go up after during cachexia. So which genes are we looking at? So here we clustered them a little bit together, and you see that like MERV, P62, ALK, and MUSA1. So these are genes related to these degradative machineries which I mentioned before, and this is in cachexia, and this is after activation of AKT. So we're not only restoring, let's say, protein synthesis, we're also inhibiting the degradation machinery. And so these were genes, and these are the processes. So actually when we now look at the protein level, we can also see that during cachexia you get this lipidation, for example, of LC3, but after, I don't know why it's missing the, I don't know, there's no bar here, but after we activate AKT, we actually don't see this lipidation anymore, suggesting that we actually are able to inhibit this increased autophagy flux, which we find in the cachectic animals, and also the ubiquitination of the proteins, which we see this increase in ubiquitinated proteins in cachectic animals. We don't see this anymore after AKT activation. So suggesting that the transcriptome analysis actually matches quite nicely what we see also at the protein level. We also, together with George Ruas in Stockholm, we did some deconvolution analysis on these RNA sequencing studies we had, and so you see a lot of different cell types which we find in these different conditions. So here you have the sham, and these are the tumor-bearing animals, and some very interesting ones. So here, for example, some anti-inflammatory macrophages which actually decrease, at least in our hands, in these C26 mice. So this is the red one here below. This I'm a bit shaky with the hands, too much coffee, but it's it actually goes down, as you see here, in the tumor-bearing animals. And after activation of AKT, we actually see that they go back up. While when we look, for example, here at these macrophages, this is actually a little cluster here. Sorry, it is what it is, this shaking. But it's only present in the tumor-bearing animals, and it's no longer there when we activate AKT. So suggesting that we have also some systemic effects, which clearly matches nicely also with the transcriptome data, which was quite normalized. And so we have these numerous positive results, actually, if we add it up. So force is restored, the transcriptome is nice, we have metabolic improvements, even some denervation effects, which were mentioned by Roberto already previously. We don't see them anymore. So it really seems to have a very positive effect. And so we wondered, so in addition to just fixing the muscle, could it also have some effect on the systemic environment? And so for this, we looked at the tumor growth, and so we took two different time points. So this is again let's say the body weight, or in this case the lean mass. We see this over time, and it starts to drop off. You see here in gray, and so we have when they lose about 5% of body weight, so they start to lose already lean mass. We start to give tamoxifen to these animals. So this is our time point T1. So when we start giving tamoxifen, so this turquoise area. And then when these control mice reach about 15 to 20% of body weight loss, then we also stop the AKT, or more or less when we have a recovery in our muscle mass. And then we took out the tumors at time T1 and T2. And this was actually quite interesting because when we looked at T1, so this is just a control tumor-bearing animal, so you see that from T1 to T2, this is actually a 5-day period, 4 to 5 day period. There's a really strong growth, so we're really in a rapid growth of the tumor. And what we found was that in our AKT animals, this growth is severely blunted. So it's a little bit higher than it was in T1, but it's definitely smaller than what we find at the end point in the control mice. And this systemic effect, let's say this effect on the tumor, we also found this similar systemic effect on the spleen. So this is already mentioned before, the spleen tends to increase a lot. So also we find about a 3-fold increase in the spleen. And when we activate AKT, we don't see this increase anymore. So suggesting that we have a very serious effect. And keeping in mind here that the AKT is really muscle specific. So this Cree driver is, you know, we know it very well. It's only expressed in the muscle, so we're not hitting anything else. So then we looked, and also Fabio already asked this a couple of times during the conference, what about histology? So just not looking at the mass, and so here we are on a bit shakier ground because I'm not a muscle or a tumor biologist, but so this is a hematoxylin eosin staining of one of these tumors, and so this have these proliferative, let's say, parts where there's proliferation ongoing, and you have some parts where you start to see some necrosis. And when we looked in the AKT mice, we start to actually see that these tumors, not all, but quite a few, they started to show these necrotic areas. So it's still they have proliferation there, but they also start to show necrosis. So really alterations in the histology of the tumor. This proliferation, we also quantified this, so we used the Ki67. I don't know if you can see it, but anyway, we counted the proliferation, examining the proliferative phase of the tumors. To see if there were any changes going on there, and also there we found a reduction in proliferation of the tumors. And so then we wanted to understand a little bit better what was going on, so we wanted to do some sequencing, some RNA sequencing in the tumor this time. And there we bumped into something which I thought, you know, it'll be a little bit of an interruption in the story, but I think it was for me a bit unexpected, so I thought I might share it here since this is the right audience. And because I didn't find this in the literature. So actually when we did these RNA sequencing in the tumor, and so you find genes going up, going down, some of the things we found which were a bit surprising were that we found like muscle contraction, myofibril assembly, which is quite weird because, yeah, this is very short tumors, right? So this is we're talking about 2 weeks. So we're not looking at tumors which are very long to generate, let's say, ex novo muscle. And initially I thought it was an error, that we just took it out wrongly. But what we did actually, we did a staining. So unfortunately you can't see it very well, but this is a whole tumor here. And we did just stain for a myosin heavy chain antibody. So actually we just, this is a fast myosin heavy chain. And we found these sporadic muscle fibers here and there throughout the tumor. So this is not a piece of muscle which was around the tumor. We found some let's say, isolated fibers here and there. And actually we went back to all our previous tumors and also some of our collaborators in the audience here. Which I hadn't told them yet. But in any case, we found these myosin heavy chain positive fibers in, I think, 80 to 85% of our tumors. So I think I put this picture here because this is a carnosus muscle. This is a very small muscle. This is our working hypothesis what it is, but I'm open to better suggestions. This is actually a muscle which is under the skin in rodents. So it's not in humans, but it's in rodents. And so I think it might be due to the fact that when you inoculate these cells, they tend to inglobate, how do you say this in English, to incorporate some of these fibers. And this is a bit casual. Some tumors has more, some have less. So it's something to keep in mind when you start to do omics, I think, on a tumor without knowing what's in there. It might, could be a bit tricky. So just to let you guys know. Going back to our story, when we actually looked through this RNA sequencing on our tumors, forgetting a little bit on about this muscle part here, what we found, and this is going back to the proliferation data, we actually found that there is a reduced expression, as you can see here, of genes that cluster together in this, with this proliferation. So supporting also our Ki67 data of this reduced proliferation. So, sorry, so yeah, this was nice, but I think there's, you know, it's good to test these things also in different models. So we also use the other, let's say, standard, if you want, Cachexia muscle, model, sorry, which is the Lewis lung. And there, here you can see, we find, in this case, when we give tamoxifen, we start to see an increase. So this is actually, we do this at day 19 to more or less keep the timing after the inoculation the same. So they are not cachectic yet. So this is not surprising by itself. We start to see some loss in fat mass. So they're starting to go that way, but the muscle are not smaller yet. Here you can see we also have in this model a nice increase in muscle mass. So again, AKT is able to also in this model of cancer cachexia to induce muscle growth. And what I think is quite nicely, we need to add some numbers here, but also here we start to see the same effect as what we found in the C26 model. So suggesting that really we are able in a similar time frame, so it's quite quick to actually impinge on tumor growth and tumor progression. And so now we are ongoing, we are in this search to see some of the potential mediators of this effect. And yeah, so this is just the overall conclusion. So, and I would just want to thank the people. So Alessia, who did, I think, most of the work, she actually left, unfortunately. She fell in love with the wrong guy at the wrong place, and so we lost her. George for actually hosting Alessia for a while, and Roberta for the collaboration, and you for your attention. Thank you.
Great talk. So FOXO transcription factor is a downstream target of the AKT. So how much of the observed effect, especially the change in the transcriptome, do you think it's due to FOXO? And do you think there is independent effect?
So the quick answer is, it's for me impossible to say what is due to FOXO, but I think Andy will show some data on MRF, which suggests that, you know, I think the degradation part, this is very likely FOXO dependent. So it's not necessarily the mTOR, but this is more AKT FOXO for me. So this inhibitory effect on autophagy, on ubiquitination of the proteins. So I think it's a bit of a mix, but I don't know how much. For sure, the regrowth is mTOR.
There was a publication, like an old one, when they target FOXO, interestingly, there was hypertrophy, which it doesn't like really given how FOXO. Yeah, but amazing work. Thank you.
Yeah, I don't know. I mean, I have seen a lot of these FOXO mice pass along over the years. I've never seen hypertrophy myself, so I don't know how. You mean when you knock it out, the muscle starts to grow?
I believe they were targeting it using oligonucleotide.
Okay.
Yeah.
Okay, so now I'm not familiar with this. Sorry.
Thank you.
So that was a wonderful talk. You know, from a clinical application, because we, as you probably know, we've been targeting AKT for a long time. I mean, it's been historic that we've been looking at AKT with LY inhibitors forever on multiple different types of cancers, pancreatic, colorectal, what have you. When you look at this data, it's obviously quite provocative, right? Because to some degree, AKT targeting hasn't worked. And so can you give me a little bit of an idea as to how to sell this on the level of, yes, promotion and maintenance of to some degree, muscle mass, in the ideas that AKT to some degree is a very proliferative marker for colorectal cancer or other cancers?
Yeah, but you're mentioning, I mean, you mean the AKT in the tumor? Yes. So now the idea here is, of course, we have AKT in the muscle. So perhaps I didn't catch your question.
So if you think of it on a systemic level,
Yeah.
right? Because ultimately one day you're thinking of systemic kind of control of AKT in the muscle. So when you're thinking of it on that level, and you're applying this in a translational clinical point.
Sure.
So if you're going to upregulate AKT in muscle, number one, how are you going to do that? And is that going to influence tumor progression? Kind of, it would defeat the purpose of what ultimately you're trying to do.
Yeah, so of course, I mean, this will not be a solution I will bring for sure. So this is a, but but this is of course an issue. I think it does latch on to exercise in a way, right? So I mean, there are different types of exercise, some which could modulate, but of course this is not as we are doing here. I think this works well as a proof of concept. Okay, I don't see immediately right now how this could be effectively translated, since even the delivery specifically stuff to muscle systemically is not, is not there as well as I know. But with these AAVs, you never know if.
We're good. One time.
Okay, so incidentally, I was going on the same point. So you know that exercise can suppress tumor growth,
Yes.
And that is mainly has been ascribed to the muscle secretome. Do you have any hints on the secretome of your mice?
No, we don't. No, but yeah, but I think that's exercise. It's aerobic exercise generally, right? What is done.
Well, take care, because aerobic exercise activate PGC1 alpha. In the transgenic PGC1 alpha tumor growth, with the models we use, the tumor grows more.
Okay. Yeah, no, but. Yeah, but so AKT, I would match it more with like resistance exercise, but this is generally limited to very small muscle groups, right? So it's not so. So I find it's difficult to imagine how this would. Yeah.
It's a quick question, great talk. So obviously AKT, or protein synthesis in the muscle, is obviously very energetically costly. Do you see changes in food intake, or are you changing the nutrients that's going to other sources, any other tissues that are changing or atrophying to compensate for the growth in muscle?
Any other tissues is quite a broad question. No, so yeah, it's a very good question. So I mean, yeah, we do see a slight increase in food intake, because you have to imagine that, so we showed, let's say, muscle mass of one muscle, but this is really all the muscles. And so this is metabolically very intense, of course, for these mice. And for example, what we would have liked to have seen is an increased survival of these animals, but we don't see this. And I think we also don't see the liver doing all too well. So I think there's a lot of compensation going on to sustain this. Partially this is food intake, but only partially, I think.
Hang on, Bert. Can you quickly speak to the fasted or fed state of your mice? I think do I remember correctly that some people see different changes in phosphorylation of AKT depending on if their mice were fed or fasted?
In general, you mean?
At the time of sacrifice.
Okay. Well, yeah. I think if you sacrifice, I don't understand exactly the question, Lee.
So when you sacrifice your mice, did you fast them before sacrifice?
No, we don't fast them, but this is a hyper-activated situation, right? So but if you look at the endogenous one, yeah, I would keep the timing constant.
Yeah, that was in your endogenous C26.
Yes.